Okay, so here we are looking at PDQ 9 on biotechnology. You're not going to be asked about ethics. You don't have to worry about this box. The applications over here are interesting, but also slightly outdated because it's a little bit old, but not part of the curriculum for AP Bio. So these two boxes on the bottom left, bottom right, are not our concern. So our concern is going to be up in the top box, up here. Okay, so there are three things I'm going to talk about. Um, Very quickly, we'll talk about restriction enzymes. Very quickly, we'll talk about PCR, and then we'll spend a little bit more time on electrophoresis. Okay. So restriction enzymes, all right, they work because, well, let's say they work on DNA, and they work at very specific recognition sites. They are palindromic only in the sense that if you look, this whole light green area here is the recognition sequence for a particular enzyme. And it reads the same back to front when you read it from 3' to 5' or 5' to 3'. So if you look at the top strand, you're going from the 3' end, C, T, T, A, A, G. And on the bottom strand, if you go from the 3', it's the same thing, C, T, T, A, A, G. Restriction enzymes are good for digesting DNA for study, and they're also good for digesting DNA in the context of inserting a gene that you want expressed. And we talked in class when I taught you about the ampicillin resistant plasmid to be transformed into bacteria to do the plates. We also mentioned what would happen if we put an insulin gene into the same plasmid. How you would do that is you would take a restriction enzyme, cut open the plasmid, okay, and take the insulin gene, which in this slide would be the dark gray area here. It's a gene, lots of DNA bases that codes for insulin, and you digest it with the same amino acid. It ends up with the same complementary ends, so that the T, the A, and the A line up with the A, the T, and the T, so that it actually fits in right here. Okay. Now, once we ligate them together, you now have a continuous strand, 5' to 3', a continuous strand on the bottom, 5' to 3'. What you have is a functional gene that you've inserted into a plasmid, and that will be expressed. So sticky ends and restriction enzymes are a way to cut things open and insert them when necessary for engineering, but also you can just digest DNA. Like, it doesn't have to be a plasmid. If we take linear DNA and digest it with a restriction enzyme, it's going to cut the linear chromosome, the linear piece of DNA, into chunks, into pieces, and those pieces will be of varying lengths. Okay, it's not a regular spacing for restriction enzyme sites, what we call restriction sites. It's I think it is random. I think it's evolutionarily random. So that means the location of these restriction sites on a particular chromosome vary from individual to individual because there's variance amongst the introns, and there's no reason to think that a restriction enzyme can't cut at an intron. So the reality is you can break DNA into chunks, into fragments of varying size. I can't say that enough times. Varying sizes, and then you'll have to separate them out. Okay. So that's it for restriction enzymes. Don't overthink it. Don't overdo it. You don't have to go any further with it. Okay, just the idea that they cut DNA to specific sequence. Polymerase chain reaction is a fun one. Okay, it's a chain reaction because it happens over and over and over again, repetitively, and it is involved in making many, 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 many copies of DNA, which means you have polymerase involved as well to make the copies of the DNA. Okay. Many copies of DNA, you don't need to use cells to do it. You can use the enzymes and a soup and a thermocycler. Okay. The idea is this. Okay. If I want a copy of DNA and I need to study it, I have the DNA. I know what the sequence is that I want to study. I know what my target is. And I want to, let's say, overnight, make 1,000,000 copies of this thing. All right. Because the more copies I have, the more easy they are to look at, the more easy they are to separate on a gel, the easier they are to study, the easier they are to manipulate. Trying to work with a single molecule of DNA is ridiculous. I need to have thousands of, tens of thousands of, hundreds of thousands of, millions of copies of that to that I have enough volume to work with. These molecules are small, so the more copies I have, the better. The idea of PCR is to take the double-stranded DNA that has the target sequence, separate it using heat, Okay, we basically open it up. We don't heat it so much that it actually denatures the entire DNA molecule. It just breaks the hydrogen bonds between the nitrogen bases from one strand to the other. So now we have two strands that are open. If I add a primer, which I can buy at a store, then I let DNA polymerase do its job. DNA polymerase can now bind to the primer, just like we learned in replication, and it will then go ahead and make, well, the copy. It's going to replicate the DNA as it is. What you're doing, though, is you're eventually making multiple copies of the target sequence. Now, look, we go from one copy up here to 2 copies to 4 copies to 8 copies. The idea is if I let this thing go overnight, I'm going to end up, okay, with thousands upon thousands upon up to millions of copies of this target sequence that I can now study. I can cut it with restriction enzymes. I know what the sequence is. I'm looking for it on purpose. These things are cool, and it allows you to make so many copies, so many copies. The cool part about PCR is that we discovered, the scientific community discovered, a polymerase that doesn't denature at 98 degrees. Because if you think about it, we put all this stuff into a tube, we plop it into a machine that is programmed to heat up, cool down, heat up, cool down, heat up, cool down automatically. Okay? And that's why we can set it and forget it. And then we can go home and come back the next morning, and the thermocycler will have gone through its program and probably done anywhere between 20 and 30 cycles. And that means you've got 2 to that many repetitions. So 2 to the 23rd power is 8 million. So if it goes through 23 cycles, you've got 8 million copies of the DNA. The reality is, needing to do that on a thermocycler so that I can get it warm enough that the DNA strand denatures. Oh, let me not. Well, it says denatures. I'm okay with it denaturing, but we're not breaking the DNA strand, the backbone. We're breaking open the double strand. The strands separate, but we're not destroying the backbone. But in order to do that, 94, 95, 96, 97, 98 degrees Celsius, somewhere in there, we also have to have in that soup nucleotides that are available so we can build new DNA. But an enzyme, the polymerase, and the primers have to be there as well. So the polymerase is the more sensitive enzyme, the more sensitive molecule in there, because it's an intact protein that we're hoping works. And what happened was they went into the hot springs at Yellowstone and found a bacterium, Thermus aquaticus, that lives in hot water, hence the name Thermus aquaticus. And they found the Thermus aquaticus polymerase. That's why it's called TAC. So the TAC polymerase is stable at hot temperatures. That's it. So you put that in with your primer, with your DNA target sequence, and the nucleotides so that you can build new molecules. You run it in this machine overnight, and you end up with millions of copies. All right. Now, the millions of copies are going to be enough that we could separate them out on a gel. So the last piece of this biotech puzzle is something called gel electrophoresis. Notice this, that we're going to separate the fragments based on their size. Okay. If we look at a gel, well, let's back up a step. What we do is we make a gel out of agar. It's basically jello. Okay, it's not very flavored. There's no sugar added, but it's basically jello. It's a filtering cross-hatched matrix of gelatin, and depending on the concentration of the gel, you have a filtering process in place. And if you make the gel really concentrated, then it's a really, really tough filter to get through. If you don't make it very concentrated, it's a lot easier to get through. But in any event, the gel here acts as a filter based on size. So if I take these test tubes and I've got DNA molecules that have different sizes. In this tube, I've got a bunch of DNA that's of different sizes. I take my PCR, I make millions of copies of it, then I digest it with a restriction enzyme, and it chops it into chunks that have different lengths. And I do the same treatment for this different sample of DNA and the same treatment for this different sample of DNA. I plop those into these three-dimensional holes. These are wells. They're basically, well, holes in the gel. I drop them in, and I run the gel using a power source. There's a reason for this. If we put the plus side of the electrical current on the far end of the gel and the negative side of the electrical current on the start end of the gel, you will pull the DNA through the filter because DNA is negatively charged. And negatively charged things are going to be attracted to the positive end of the power source, to the electrical current. That's because opposites attract. So DNA's nuclear charge is negative. That way, even if it's not in the nucleus, that was a silly thing to say. Even if DNA is not in the nucleus, even if I put it in a test tube, DNA is negatively charged. So when I drop it in the gel and apply an electrical current, it'll pull because of electrical force through the filter. Small strands go further, larger strands don't go as far. So this gel was run from left to right. The strands that are staying near the well, these are the bigger ones. They have a tougher time getting through the gel filter process, the matrix, and the bands that form at the bottom, or further away, I should say, are the smaller molecules, the shorter chunks of DNA. And you can compare the patterns of different things. It could be a suspect at a crime scene, it could be a paternity suit, you could be just running plasmid studies using different restriction enzymes and realizing that some plasmids have more than one restriction site. You could look at a linear chromosome and realizing it has multiple restriction sites, but one person might have 5 introns, restriction sites within the introns. Another person might only have 2 because introns can be different amongst different members of the same species. That reality leads to sort of a mixture of things going on here. So you compare them. This is called a DNA fingerprint. Okay. Last piece of the puzzle for this little brief talk on biotech is that you have to do something with the DNA so that it's visible. These happen to be treated with ethidium bromide, which glows under a black light. So fluorescence or radioactivity, so you can develop it on an X-ray sheet to give you basically an X-ray photograph of what's going on. There has to be some way that the DNA is visualized, because when you drop it into the gel, it's an invisible molecule. It doesn't have density enough to just show up or be darker. Some labs have developed a technique where the original DNA that you drop in the well is actually stained already, and that's a possibility too. It's just less reliable, so that when you pull through, you can actually see the dark bands, but that's less reliable. So what we often do is use radioactivity to make them glow under a certain source, or fluorescent, so they glow under a black light. That way we can visualize the DNA. And that's how we compare it. Okay, so the DNA fingerprint is unique, but other than that, it's a gel, an electric field. DNA moves to the positive because it's negatively charged. Different sized fragments move differently. Smaller fragments move faster through the gel, which means they move further over a period of time. And then we have to be able to visualize. Okay? That is it for what you need for tomorrow's exam, okay, for the midterm. All right. I'd like to spend more time on it. We might pick this up again after the midterm, just because some of this stuff deserves a little bit further of an explanation. But these few minutes on video will get you through the exam. There's only 1 or 2 questions about this on the entire exam, so don't overdo it. Just hit the highlights like I did here, listen to it once, look at it twice, and be done. And hopefully that'll work for you. Take care. Good luck. I'll see you tomorrow.